So thank you very much, everyone, for coming to our weekly Innovative Discovery series. We are absolutely thrilled today to have our friend and colleague, Dr. Julia Barthold, from AI DuPont Hospital for Children, to talk about studying genetics of complex disease. But before I introduce our speaker, let me go over some housekeeping issues. Please make sure that you sign in, and please RSVP so we can better prepare for the next session. Next session will be smoking cessation and relapse prevention during pregnancy by Victoria Coleman-Koger on July 15th, followed by the utility of alternative article-based metrics for clinical and public health researchers by Margaret Chisholm on July 22nd. Also, do not miss the next TED Talk on July 7th. Pan Wu will feed your mind, our mind, with mediation analysis, how exclusive breastfeeding affects child obesity at age 4. The full schedule is on the Axel website. All the previous presentations are available on our website, DE-CTR.org. So let me go back to Dr. Barthold. Dr. Barthold is currently Professor of Urology and Pediatrics at Jefferson Medical College. She's also the Director of Research in the Division of Urology at AI DuPont Hospital for Children and Affiliated Professor in Biomedical Engineering at University of Delaware. She got her MD from Northwestern University Medical School, where she also did a residency in surgery and urology, and further training in pediatric urology from Wayne State University School of Medicine. She has had multiple awards, and in 2013 was awarded the John Duckett Pediatric Urology Research Excellence Award from the Urology Care Foundation. She has been the PI on multiple NIH grants and recently completed a very large R01 related to the genetic basis of cryptorchidism. She is also the Nemours PI for our Axel CTR grant, has been on multiple committees, and also is currently the president-elect of the Society for Pediatric Urology. Congratulations. She has a very large number of peer-reviewed publications and book chapters. Thank you very much, Julie. We are really excited to hear all this stuff.
Thank you, Claudine. Appreciate it. So I wanted to talk about this just because of my experiences as the PI of a grant looking at Cryptorchidism, which did turn out to be a complex disease. And I would like to try to present this in a way that people can understand basic concepts about complex disease. And also, I'm going to focus somewhat on genome-wide association studies, as well as beyond GWAS into things such as systems genetics. Please feel free to interrupt me if you don't understand anything. I'm going to try to go more slowly than I usually do to make sure that we understand all the concepts. So first of all, I am going to talk about concepts and tools that can be used for studying genetics of a complex disease. But I'm then going to talk about my experiences in human GWAS and an animal model of cryptorchidism, or undescended testing. So as we know, the human genome is quite large and comprised of A, G, C, and T nucleotides. Sorry. Is there a pointer here? No. Okay. 23 chromosomes, 22 are autosomes, and the X and Y chromosome determine sex. And the number of genes in the genome has gone down gradually and is now estimated at 20 to 25,000. But only as much as 1.5% of DNA is actually coding. So it used to be thought that most of the DNA, besides this, was quote-unquote junk DNA. But with very extensive studies looking at transcription factor binding sites, areas of conservation and enhancers, etc., throughout the genome, through the ENCODE project, it's now known that a lot more of the genome is functional. So why are we interested in genetics? Because We want to understand the cause of disease, and that will help us design biomarkers, look for therapeutic targets, and prevent disease. But we also want to be able to predict how individuals will do, which is, as you know, considered personalized medicine, which is becoming more and more important in this day and age, and supported by the president, where we will be better able to diagnose, treat, and prognosticate disease. So this is just a recent review of some of the more common diseases where heritability is known. And what heritability means is the portion of phenotypic variance or trait that's attributable to genetic factors and is determined from family and population studies. And as you can see, many of the common diseases have a substantial heritability, meaning that there are genes that are contributing to them, but the vast majority of these are polygenic or due to multiple gene defects or variants. The classic Mendelian or monogenic traits are single gene defects that have a large effect. In other words, they're mutations that actually change a protein significantly, but they can be influenced by other modifier genes. And as we know, the types of protein dysfunction is related to missense and nonsense mutations that significantly change the quality of the protein, insertions and deletions that can cause frame shifts or stop codons, and also splice site mutations that affect the actual transcription and translation of the full protein. On the other hand, complex traits are by definition phenotypes that are influenced by multiple genes, and in addition, they interact with environmental factors. And the genetic variation comprising complex traits is very broad. And to make it more difficult to identify these genes, each locus has fairly weak effects in the 1.1 to 1.5 fold range. What has been found is that typically these traits are associated with non-coding variants, so that they may be the actual causal variants may be at transcription factor binding sites that affect binding of a protein to a part of the DNA to help or to stimulate or repress transcription in enhancers which are similar, and the variants may also alter DNA or protein stability, or alter things like small RNAs that bind to mRNAs and regulate the levels of RNA message in the cell. So when we're looking at genetic variation, the most simple is the single or simple nucleotide polymorphism, or SNP. Indels are small insertions or deletions, and copy number variants, which in recent years have become more and more interesting and studied more and more, refer to variations that end up with causing greater than 50 base pairs of insertion or deletion. And recent studies show that this is not uncommon at all to have copy number variation within everyone's genomes. And in fact, as many that they found at least 100 genes that can be deleted without any phenotypic effect. So that makes the search for the genetics of disease more complicated, just because it's not clear that which variants may have true functional effects. So many for CNVs, the state of the art is to determine whether it's benign or pathogenic based on healthy populations or other family members that don't have the disease of interest. So this is just a depiction of the association between rare and common variants. On the left, that you see rare variants that if they have a large effect size, they are easily identified as causes of Mendelian disease that are inherited within families. And common variants are typically have a lower effect size, and these are the variants that we're looking for when we do genome-wide association studies. So what are the tools that are used to identify genetics that are associated with particular diseases or traits? The one that's been standard over many, many years is linkage analysis, where you have a family with multiple members having the trait or disease, and you genotype family members either using restriction or macrosatellite markers, and identify how the trait is being inherited in association with these markers, and identify loci on the genome associated with the disease. When I started becoming interested in this, the only thing really available was candidate gene sequencing. And so basically, you know about biological pathways of your disease or your target tissue, and there are animal models, knockout mice specifically, or there are also some spontaneous animal models, particularly rats, that so that you sequence the entire gene, the expressed portion of the gene, the exons and the intron-exon junctions, to look for variants that could affect the function of that gene. More recently, genome-wide association studies came into widespread use, and these involve arrays of typically 300,000 to 2 million SNPs, where each individual is in each individual's DNA is genotyped across all of those SNPs, and the other thing that, as part of that genotyping, there are programs that actually can determine copy number variation within those individuals and compare cases and controls. And these are case-controlled studies, so it's very important that the cases have a very clear phenotype, and the controls are also appropriate in that that you know that the disease is not going to be present. So, for example, in my disease, I'm studying undescended testis. I use male controls because the female, we don't know what the phenotype would be in females. What GWAS can do is provide fundamental insights into genetic architecture, and in fact, with a lot of collaboration across institutions, so far it's identified thousands of genome-wide significant signals associated with many, many traits. So what's important about GWAS is it's unbiased, unlike candidate gene analysis. And so things that you find are going to, it's not hypothesis driven, but it can generate hypotheses. And what's important to know is that the markers that are on a SNP chip board to do GWAS are often not causal, but they are in linkage disequilibrium with a causal variant. They may be causal, but most often they're not. And so once you know what portion of the genome you're interested in, you need to look at other variants in that area. The power to detect an association in GWAS is based on multiple factors, but most commonly sample size. The samples need to be quite big, and power analyses are done based on the expected effect size for variants. And then whether or not you're going to find anything in a GWAS is really very dependent upon the genetic architecture of the disease. How is it associated with common variants or not? If it's associated with rare variants or it's very complex and heterogeneous, you're not going to find much unless you have very, very large populations. So again, GWAS is based on what was a common variant, common disease hypothesis, and which I'm not sure. It certainly holds true for many diseases, but certainly many do not fit into that fit with that hypothesis. But what we do know is that when we're doing these studies, we have to use a very stringent p-value cutoff for genome-wide significance because of all the markers that are being tested. And so in order to correct for multiple testing, the p-value cutoff is 5 times 10 to the negative 8. And so when you do a study, you may only find a handful of genome-wide, or even one genome-wide associated locus that's proven, where there may be many others that you're not able to prove because of the limitations of the methodology. And those, what we do know is that these significant signals that I'm talking about often do occur outside of causal or obviously causal regions. And in fact, the majority of them map to regulatory elements within the genome, which is interesting in and of itself. The GWAS doesn't provide any information about rare variants, and some diseases are associated with both common and rare variants at a certain locus. The other issue with GWAS is that you have to have a homogeneous population because allele frequencies, or the percentage of a population that has a particular allele at a certain variant, is going to vary depending upon their ethnicity and race. And so you have to account for this possibility of population stratification. And if you're looking for low effect loci rather than, which is what we often find in complex disease, you do need large sample sizes. And even then, it's not unusual for the SNP not to replicate in another population, which is another requirement to consider that SNP definitively associated with the disease.
So when you talk about large sample sizes, it means the order of
thousands.
Oh, thousands.
Thousands, 10,000s. Yeah. And it depends on the disease, obviously. Mm-hmm. So there have been, and I'll talk a little bit about this more. So pleiotropy is something that contributes to complexity of genetics, where, and it refers to a situation where genetic variant actually causes variable phenotypic effects. As it applies to GWAS, it's called cross-phenotype associations. That means that you can find significant association of genetic variants at a locus that are associated with multiple traits or multiple diseases. And this is in support of prior epidemiologic studies that actually show certain diseases have shared risks. But the problem is that it's unclear sometimes what exactly, where the causal variant is. If you're looking at a whole region, it could be a C and V that you may or may not be able to interrogate, and it may just involve a pathway. And so if you do see cross-phenotype associations when GWAS, it may or may not be true pleiotropy. So this is sorry I don't have a pointer, but this is just an image of different types of interactions between what would be considered a true causal locus, which you may or may not know in the red star here. The SNP that is shows significant association in a GWAS. So in the biological case, in A, you see that region is associated directly with two independent phenotypes. However, there may be two different causal variants at the same locus that cause the different phenotypes, or there may be those two variants may not be in linkage disequilibrium or inherited together based on recombination, but yet in the same gene. And then it gets more complicated from there, where the phenotypes interact with each other, or where there may be spurious results that are related to misclassification when you're choosing cases and controls, and how the methodology used for a specific study, or where causal variants are close to each other but actually in different genes. So I think one thing that there's not much about this in the literature, but there are increasing numbers of studies that are using alternative ways of genome-wide association that are focused on something other than genes. And PWAS is phenome-wide association, which is sort of the opposite of what GWAS is. So and this has become more of interest because of all the medical record data that are available. So you can look for a person's set of traits or longitudinal information about them over time and look to see which genetic variants or set of variants is associated with their traits or with multiple traits. So this is actually something that I think will become a lot more widespread in the future. The other thing that, of course, is often not considered in these studies is environmental effects, which are difficult to measure in certain populations, but they certainly contribute to the risk of complex disease. But if you have information over time from individuals, you can, in some ways, test environmental effects or life stages and the appearance and disappearance of specific traits. So what has been debated for a number of years about GWAS is that when you do a genome-wide association study and you find variants, they often only account for a very small percentage of cases of the disease. So you may find, for example, I'm going to talk about a hypospadias GWAS. They found 18 variants and they accounted for 9% of the incidence of the disease. So there's this concept of missing heritability. What is the cause, if not these loci that are found by genome-wide association studies? So as I showed you in that prior table, we do know what the heritability is for many diseases. So what is causing the disease other than what we're finding in GWAS studies? So the possibilities are that there are multiple rare variants, which rare being defined as occurring in greater than 1% of the population. There can be gene-gene interaction or epistasis, which is difficult to model when you are studying GWAS data. You need large numbers of genotypes to be able to do that, but it's possible. That there can be shared environment in families, which could look like a genetic disease, but it's more of an environmental disease. And as I mentioned, CNVs are not routinely interrogated using GWAS. They're more commonly looked for in syndromic diseases using comparative array hybridization. And then there's epigenetics. And I think this is an important thing to consider because we know that it's not just the sequence of the genome that's important in terms of what your phenotype is. If you look at identical twins, they have the same genome, but they have differences as well. So there are secondary effects that alter gene expression throughout life. And some of the changes in the epigenome can occur very early on. And this is a hot topic of research, so that things like environmental toxicants, nutritional factors, and social experiences, especially early in life, can cause long-standing changes in the epigenome, which, now I'll talk a little bit more about what that specifically is, but this is a very interesting study that people may have heard of the Dutch famine, where there was a methylated region in the insulin growth factor 2 gene that showed reduced methylation only after early but not late gestational exposure to people who were fetuses in early gestation during the German food embargo after or in late World War II. And so this had long-lasting effects that occurred to at least middle age and had effects on growth and probably other phenotypes that we may not even measure. So what is the mechanism? Most commonly, it's methylation at CPG islands or sites that occur in promoters, but they also occur in many other places in the genome, including within genes and in introns and in exons. And when the cytosine is methylated, there is a tendency to silence gene expression, and the demethylation tends to allow gene expression. There are also histone modifications which affect how chromatin assembles. And as you may recall, if chromatin is condensed, the gene expression is inhibited when an open chromatin allows transcription to occur. What we know is that any epigenetic reprogramming that occurs in cells of an individual is reprogrammed during embryonic life. And then as the embryo develops, those areas are remethylated based on cell type. And that's why you go from an embryo with very few cells to a multi-organistic individual, because each cell has a different gene expression pattern. And because of that, epigenetics is tissue specific and is based on not only tissues but cells within those tissues as well. There are some studies that suggest that transgenerational inheritance can occur of epigenetic phenomenon. And this is mostly in animal models, although there was a study of women exposed to DES whose grandchildren were more likely to have reproductive abnormalities. And so but the mechanism of that was not shown. But in animal models, the idea is that if you expose an animal who's pregnant to a particular toxicant, and the ones that I know most well are reproductive toxicants, so they cause reproductive abnormalities in the fetus. That the offspring of that dam will, if you continue to mate into subsequent generations, you can continue to see abnormalities over multiple generations, which means that it is, unless you're changing the sequence of the DNA, which is not what is known to occur, it's associated with their actual methylation changes in the genome. And so some of these regions may escape reprogramming in the fetus. Another way to help understand complex diseases beyond GWAS is systems genetics. And I'm just going to mention this, and I want to mention that the person who has written the definitive review article on this is Mehdi Svelik, who was here interviewing at University of Delaware, and I met him a year or two ago. He's now in Charlottesville, and he's coming November 7th to give a talk at Nemours. So we'll send that out to everyone. But the idea is that if you have multiple genetic variations, and you're going to have altered gene expression and protein expression and metabolite expression, and then this can be expanded to include things like gene-gene interaction and gene-environmental interaction. But this is such a wide area of study that rather than just focus on specific loci within the genome, the idea is to look at biological pathways and more general effects of diseases and go backwards from there. And so and basically what it does is help understand the molecular basis of complex traits, which can then inform further studies that are focused specifically on gene candidates. The methodology is identification of expression quantitative trait loci, or EQTLs. And what these are areas, or these are genes are genes whose expression is altered by a specific variant in the genome. So a SNP is associated with a variant in a local gene, or it could be something that is well downstream. They're either called local or cis, or distal or trans, and some of these trans effects can occur even on different chromosomes, so it can be complicated. But the way to study this is to measure, is to look at a population, genotype them across the genome, and then compare the variants at specific loci with expression of genes. And there's a computational way to do this, with which Mehdi is well versed in. This has become important because there are now there's a huge database online, the GTEx Consortium, that contains multiple cell types and multiple tissue types, and compares gene expression with variants in the genome. And again, this is tissue specific. So now I'm going to talk a little bit about what I did with studies of undescended testes, which is a common disease, one of the most common birth defects that occurs, but of course only in boys. And there was some evidence that there's moderate genetic susceptibility, but also environmental effects as well. So that suggests heterogeneity. And on top of that, it's a feature of hundreds of syndromes. And so we know that it's likely polygenic. It is also part of what's called the testicular dysgenesis syndrome, and which is theorized to be due to the effects of endocrine disruptors. And these are environmental chemicals that we're all exposed to. There are thousands of them. Many of them affect hormone pathways, and so they can affect a fetus during development, particularly male development, but also certain aspects of female development as well. A previous GWAS of the testicular dysgenesis syndrome was really underpowered, but didn't find any significant loci. But interestingly, a hypospadias GWAS that I just mentioned earlier, which was well powered, as you can see with 1,000 cases and 5,400 controls, did find multiple genome-wide significant loci that were replicated. So that was a powerful study. What we know about inheritance of cryptorchidism is that it's not a terribly high heritability, but there is evidence that that monozygotic and dizygotic twins have similar diagnosis, but the monozygotic twins are more likely to be treated for the disease, suggesting that the ones who are diagnosed often resolve spontaneously. So there's a clearly, because of the fact that the monozygotic twins are, the concordance between the two is not that high, it certainly suggests that there's an environmental effect that is significant in the disease. And so what we did is we proposed a Caucasian-only surgically proven cryptorchidism study and used non-syndromic controls and limited them to older males so that they knew they were not developing the disease and or had not developed it by that time in life. And the analyses that we used are shown here. Because we did this over a period of time collecting samples, and with changes in the genotyping platforms that occurred during that time and the availability of controls, we ended up having 2 case control groups, which complicated it. But what we did is, what's now an option is to impute genotypes across the entire genome based on existing genotypes from the 1,000 genotype database. And so once you do that, you get much better overlap between the 2 sets of genotyping platforms. And so we did that and then performed a meta-analysis of the 2 groups. And then we also, I'm going to show you a little bit about correcting for population stratification. And also, we did see an ND analysis and didn't find anything that was replicable, or that was even validated. So there are a lot of quality control steps that go into doing these studies, and so you need to make sure that the data are good and exclude any data that are not, including data with increased genotype, missing genotypes, or heterozygosity out of the range of 3 standard deviations. Anyone who appears to be relatives, brothers, father, son, can't be included. And in our data, we actually found about 1 to 2% of individuals had XYY, which is Klinefelter syndrome, which is associated with the disease, and we excluded those because we assume that the disease is due to the sex hormone abnormality. And there are other computational exclusion criteria specifically for SNPs as well, as you can see here. So when we looked at our samples, and as you can see on the bottom, the cases are on the left, the controls are on the right. We did multi-dimensional scaling analysis, and what you do is use the Stanford Human Genome Diversity Project, as you can see in the upper left, and do principal components analysis. And what it shows is that even in Europe, even for Europeans, there is quite a spread between different parts of Europe in terms of their genetics. And so if you include if the differences between your cases and controls are such that there is a significant difference in the populations, it will skew the results of your analysis. So in this case, we removed anything that deviated from the first or second components of the PCA by more than 3 standard deviations. And we found that at least for the first component, there was a significant difference between our cases and controls. And as a result, we needed to include and chose to include the first 2 components of the PCA as logistic regression in our association analysis. So of course that reduces power. And then in the end, we had 912 cases, and over 2,900 controls, but lost a number of them because of these QC steps. And we defined top signals as those with p-values less than 10 to the -5th within 100 kb of a gene. And in our full analysis, our full meta-analysis, there were 20 loci, but none of them were genome-wide significant. And again, we performed a subphenotype analysis and in fact found a lot of additional loci, and in fact many of them were with the severe forms of the disease, such as bilateral occurrence, occurring more proximal, like in the a testis in the abdomen as opposed to a testis more distal, closer to the scrotum, or occurring earlier in life. And we used a more stringent threshold to correct for multiple testing. And we did find one genome-wide significant SNP, which is associated, it's near a gene that's associated with the disease, a syndrome that includes cryptorchidism. So bottom line is that we were not able to, in fact, find the multiple genome-wide significant variants that were easily found with more controls, but a similar number of cases from a study that was performed in Europe. And part of it may be that the population in Europe is more homogeneous, and that it was therefore easier for them to find association, significant associations. And again, many of our candidates of less significance, which we term suggestive signals, and we only looked at these to be able to do a pathway analysis, which I will show you. So, and we had a fairly small population of Swedish replication samples, and we're really unable to find any convincing replication, although there was some suggestive replication. So bottom line is that this is not a disease that we're able to identify any clear genes associated with, based on common variants that are identified by GWAS. So just an example of the heterogeneity. So we looked at transforming growth factor beta receptor 3 which is an interesting gene that's expressed in the testicle and is involved in hormone production in the testicle as well, and had been shown as a suggestive site in the previous genome-wide association study of testicular dysgenesis syndrome. So we looked at that and found that interestingly, this was true also for our two subgroups, but I'm just showing the data based on phenotype here, where you can see that the most significant SNP, which is shown in red at the top, and the more red the marker, the higher, the most significant SNP is taken as baseline, and all the other markers that are in linkage disequilibrium, or LD with that SNP, are red or pink. And then the areas that you can see here, the blue lines, are areas of, between the blue lines are areas of recombination. So what we found for this gene is that for a certain phenotype in a certain subgroup, we found significant, not genome-wide significant, but the most significant variants were, or suggestive variants, were at one end of the gene, and for the other group and for the less severe phenotype, we found that they were upstream of this gene. So this is just an example of how difficult it can be. We then went on and did some pathway analysis, which has been reported in the literature as a way for looking at lots of suggestive but not genome-wide significant variants that are associated with the disease, to try to get an idea of what pathways may be involved in the cause of the disease. And there are multiple software programs that do this. And in doing this, we used multiple testing correction for all, and used the syndromic genes that I mentioned earlier that are known syndromes that associated with genes and with the Cryptorchidism phenotype as a comparator. The other things that you can do after GWAS are to do gene-based analysis, and one example of that is the Vegas program, as well as use other databases that are available that will actually show whether or not your SNPs are in regulatory regions. So when we did pathway analysis, we found areas of overlap that were involved with developmental signaling primarily, and with cellular development and organization, as you can see, and that the significance was greater probably because of the greater number of SNPs that were in the group to the far right column, which includes all variants that were that had a p-value of less than 10 to the -4th. And so some of these are suggestive of the potential for androgen receptor signaling. And I'm not going to go into this in detail, other than to say that there are many cytoskeletal proteins that are androgen receptor coactivators. And in this this figure here shows that they have this LLXLL motif, LXXLL motif that is known to bind to nuclear receptors. And so we did find some variants within, actually within genes that are shown there, shaded as gray, that were suggestive signals that encode proteins that may or may not be involved in androgen receptor signaling. And so this is just a hypothesis that's based on what we've done, and it will need a lot more work. The other thing that was interesting is that another gene that's very similar, or protein that has similar structure in this region, is the SYNE2 gene that you can see there in red. And not sure. There we go. I'm sorry. And this was a gene that we identified as being associated with cryptorchidism in rats. So I have a rat model of the disease, and on the left is a normal 3-day-old male, where you can see the testes have descended into the scrotum, and the abdomen has been injected with blue dye, so you can see, there's a wide open connection between the abdomen and the scrotum in rats. And on the right side, you can see that the left testis, your right, has not descended, and the sac into which it should descend is actually laterally located, and that's what the phenotype is in these rats, which is very similar to humans. So in order to find the genes in the rat model, which is another way that you can inform search for the causes of complex traits if you have animal models, what we did is we crossed the affected strain, which is in the top right, an affected male with a polymorphic female from another strain. So we tried to identify the most polymorphic rat strain, the one that's farthest away from Long Evans, which is the parental strain of our rats. And then in the second level here, in the F1 generation, the offspring are back-crossed to females and males of the affected strain. So you're enriching for the affected DNA. And then in the F2 generation, you generate affected males. So the number of males that were affected in that second generation down is only 15%, which is much lower than the 60% that we saw in the general population, suggesting again that there are multiple genes involved. And in this case, we used we actually used a rat-based SNP chip, and we also used microsatellite markers across the genome to identify the association of the phenotype in the affected F2 generation with regions of the genome. And this is where we identified that SYNE2 gene and found an exotic deletion in that gene. So, but again, we are seeing multiple and other possibilities here, which makes it very complex. So in conclusion, the disease that I study and many other diseases that I know many of us are interested in are very genetically complex. And on top of genetic heterogeneity, if you have environmental effects, it can be very complex in order to identify areas of the genome that are associated in the absence of many individuals. But you can develop hypotheses based on this, and these are things that we are continuing with our animal model. And in addition, we are planning to do EQTL analysis for our human data set. So GWAS has a number of limitations. It is definitely helpful in providing information about genetic architecture that is disease specific, but it can help you, if you take it farther, it can help you look at pathways and biological systems. And ultimately, I think there will be more and more interest in these regulatory variants that have tissue-specific effects on gene expression in the future, and that these will help to explain some of the missing heritability that I talked about. And again, this has been many years of work that I've showed you about my own work with many people engaged from multiple institutions. And I thank you for listening. Yes?
So your rats with cryptorchidism have normal fertility, or do you have to do a lot more crosses to
They're fertile for a while, and as they get older, it's like humans. When they're bilateral, they are less fertile.
So they were fertile enough that you could do all these studies without having to do a whole bunch of extra.
Yeah. They have fewer offspring. It's not as non-syndromic as you think. Now I realize they're smaller. They tend to die in the newborn period. They're mean. I have a colleague who is a neuroscientist who worked with them once and swore that they had, a double appeared in a cave or something when he was perfusing.
Is
So
that
they may have
is that pleiotropism from this, or are there other mutations that sort of hang around with it? The differences between those rats and the others.
and
Is
outbred
it
rats.
one gene, is it physiologic effects that you're talking about?
I think it's multiple genes, and so if you look at the genes that we did find, the variants that we did find, they are minor alleles. And so what's great is that all these databases are incredible that are available now, but there's a rat genome database that has the genotypes of all different strains, and we actually put our data into that database. You could pull up the data and compare the frequency of each allele, and I didn't find any allele, and I did this in collaboration with the folks at DBI, Sean Polson, and his crew worked on this and did a great job for us, but showed that and the data are very reliable because they're very similar to the variants that are found in other rat strains. So there were no de novo variations that I found. These are all variants that exist in other rat strains, but not in the same combination. So, which is why I think, and the disease, I didn't go into in detail, but it's incompletely penetrant as well, which makes it difficult.
So it's overwhelming, I think, to most of us. So things you didn't talk about the steps from GWAS to next generation sequencing. Also, one of the things that people talk about is rather than doing GWAS, doing more limited approaches, looking only at introns.
Right. So I think what a lot of people are doing whole exome sequencing, and I didn't want to talk a lot about the methodologies because and we can always probably get Aaron Crowdy to come back and talk about that. But I think that's being used routinely for syndromic, and you may want to speak to that, Lou, but if there is a very penetrant phenotype or much more, typically more severe than what I'm looking at, whole exome sequencing can be very helpful, particularly looking for variants that are shared by individuals from different families. And I think obviously when it becomes more cost-effective, whole genome sequencing will be the next step. The problem is that all the data, that it's extremely computationally intensive. And we still don't know the meaning of all these variants, although that's going to become more and more obvious over time when more of the GTEx data are available, where we get to the point where we know what each variant, whether it could be regulatory, what gene it may be regulating. I think that's going to help with interpretation of studies over time.
So let me ask another question about epigenetics. Presumably, most of the time the gene modifications are functional. After all, things need to turn on and off genes, otherwise the whole tissue looks the same. And so it's a big fashion. We're hearing about this all the time, but I think it's hard to get a sense of, maybe there isn't an answer to it yet, of just how important this is in human disease. Most of the problems related to genetic variations in the SNPs, or more than a small amount related to epigenetic modification, or do we not even have a sense of this yet?
So regarding that, there's a couple papers recently that say the third generation of the Dutch people, who were fetuses during 44, 45, their grandchildren are shorter and more obese than the other Dutch. Now that's pretty, I mean, it's not sort of not a great study, but
maybe environmental factors there are related to that.
Well, like you were asking, the epigenetic effects can maybe transmitted for generations.
And they've shown that in animal models, but it's not. It hasn't been as clear in human.
But then the other point is, even leaving aside the inheritance across generation, there's still epigenetic modification that goes on in individuals.
Right. So,
and that's probably more important, realistically.
So it's different.
How important is it in human disease, really?
I don't think it's known. I think it's hard to do gene-environment interaction studies because they require huge numbers of individuals because of all the variables involved. the best way I think to look at epigenetics is to look at twins that don't have, that are discordant or disease, but that have the same. So you are ruling out any variation in the sequence.
But that doesn't have to be genetic. That could be pure environment too, right?
One would assume it's epigenetic. Yes.
Or
Any differences would be epigenetic.
But their environment be different.
Well, the environment has to work by affecting a gene.
Well, does it?
Well, usually.
That's the assumption. So that's as far as I can go on this.
That's true.
Yeah.
I was really interested also by what you said about the phenome. And,
Yeah.
Of course, the association with the electronic health record.
Yeah.
Right here. But so how do you see that over the lab of a person having at regular interval blood samples or something to be able to look at the genomes and the modification of the
I think
gene expression?
potentially, yeah. And that could be, I mean, if you're looking for changes in methylation over time, but obviously once you have a baseline sample, you're going to have the sequence of that individual. But you could look at their exposures over time or periods of, for example, in children, we don't know what their long-term risks are going to be, so that the ability to follow people over time and know what diseases they develop, what their baseline sequence is, and again, looking for variation in the epigenome like it is, would be a big step forward. Thank you.